Okay, so for our next discussion, we will be discussing the enzymatic activity. So what is actually an enzymatic activity? So when we are discussing enzymes, there are also factors affecting this enzyme activity. So it's not just one, but a lot. So we will be highlighting four. But first, let us define what is enzymatic activity. It is actually the rate at which an enzyme catalyzes a chemical reaction. So when we say rate, so what comes into your mind when we say rate? So we have the relationship of our factors. So we'll be seeing graphs. So I'll be showing to you the graphs of the enzymatic activity and how it is affected by these four factors, namely temperature, pH, substrate concentration, and enzyme concentration. So we'll begin here for the temperature. Okay, so how does temperature affect enzyme activity? And it is shown here in the graph. So when temperature, and take note that we have in our x and y axis the temperature, so increasing from left to right and from bottom going up, so the reaction rate is increasing here on the y-axis. And when the temperature increases, so the molecules, both the enzyme and the substrate or our reactants, they will actually, so what happens when there's a high temperature? So when you increase the temperature, they move faster, they bump into each other more often. And that means that what? The enzymes has more opportunity to bind with the substrate and the reaction will be faster or it speeds up. So as you can see in the graph here, we have the optimum temperature and we have the increased number of enzyme substrate collision. That's why it's increasing here on this side of the graph. And we have our optimum temperature. So what does the optimum temperature mean? It actually means that each enzyme works best at a particular temperature. So for example, for human enzymes, this is around 37 degrees Celsius, which is typically our normal body temperature. So what happens if it goes beyond, so increasing man ang temperature. So if it goes beyond the optimum temperature, the enzyme structure, it can start to unfold. So mag-increase ang temperature or change shape. And when that happens, the active site where the reaction actually takes place, so it will no longer fit the substrate properly. Since there is a change in the shape, there's unfolding, and the enzyme stops working well. So in conclusion, if you have moderate heat, so going to the optimum temperature, so there's more collision and there's a faster reaction. And too much heat will actually cause the enzyme structure to break down. So going beyond the optimum temperature, and the structure or the reaction will slow down or it eventually stops. So that's how temperature affects our enzyme activity. So how about for pH? So this is our representation for pH. So as you can see for our x and y axis still we have the reaction rate and here we have the pH. So let's take a look at this. When the pH changes a lot, so when you increase it, slowly increase, so from what an enzyme prefers, so the enzyme's shape can change or even get permanently damaged. So this is the optimum pH, so a maximum rate here. And if it goes beyond your optimum pH, the enzymes, which rely on their three-dimensional shape, will work correctly at this optimum pH. So this is actually each enzyme has a particular pH. So it's not the same for all enzymes. So for example, at this pH, the enzyme is in the right shape and the charges on the amino acids are correct and it can bind to its substrate well. So a common example or general example of this is that most human body enzymes, they prefer what we call the optimum pH, a nearly neutral pH, as we have discussed in the buffer or the water topic. So it's nearly around 7 to 7.5 at this range. But there are actually exceptions. So for example, enzymes in very acidic environments. So what do you think are those enzymes? So we know that in our stomach, so it's actually an acidic in terms of pH. So it actually works best, these enzymes in our stomach, they work best at pH 2. And some enzymes, they actually work best in a more basic environment, wherein, for example, we have enzymes in the small intestine. So in the small intestine, there is actually an environment where the pH is at 8, and these enzymes will work better at pH 8. So it's not very high, but it's slightly basic. And for those pH and acidic environments, so there is actually a slightly acidic environment for them. And if you move too far away from the optimum pH, so you can either move it to too acidic or too basic. So the enzyme's shape, it, it actually, its active site, it gets messed up. So it can't bind its substrate or catalyze the reaction well. In other words, if there is a big change in the pH, the enzyme structure will be disturbed and there is low activity or no activity at all. So that's how the pH affects our enzyme activity. Okay, for the next one, we have substrate concentration. So you can see it's a little bit different ang iyahang pattern in comparison to our first diagram. So we have here the substrate concentration plotted against the reaction rate. So how does this affect our enzyme activity? When the amount of enzyme stays the same, so you start to slowly add. So from low amount, we add our substrate concentration, and adding more substrate means that the enzyme has more to work on, or it needs, or you have plentiful substrate being present in the environment where the enzyme is. So this can eventually speed up the reaction. That's why it's increasing. But there comes a point where the enzyme molecule is already busy. So busy na siya, or it means that the active site is full. So no matter how much we add our enzyme, I mean our substrate, so even if you add more substrate, the rate doesn't go up any further. And there's just a small increase here. But as you can see, at some point it goes constant. So the state is what we call, take note, saturation or enzyme saturation. It's just like in solution. So at saturation, you reach the maximum rate of the reaction for those enzyme molecules under this condition. So here you have reached the maximum amount of substrate in order for the reaction to occur further. And that's how the substrate concentration affects enzyme activity. So we will now go to the last one, which is enzyme concentration. So as you can see, there is a steep increase here. So when an enzyme is not used up in the reaction that it helps or reacts with, so it actually binds to the substrate. So it helps convert it into a product, and then it helps it free, or it's free to help again. It's like a process or a cycle that it catalyzes the reaction, and then the the substrate is released, and then it can help with the reaction again. So if you keep the amount of substrate the same, so as we can see here, we have enzyme concentration, reaction rate. If there's enough substrate and you add more enzymes, so you add more enzyme in the system, you will actually increase the reaction rate. Because why do you think this is so? So it is actually because more enzymes or molecules are of course present or there to work on the substrate. Since we have an abundant amount of substrate, so increasing the enzyme concentration will also increase the rate of the reaction or the reaction rate. And this will only happen, take note, if the substrate is plentiful or present. But if the substrate runs out or becomes limiting, adding more enzymes will no longer make a big difference in this diagram. Okay, so those are the four factors that we are going to highlight in terms of how they affect their enzyme activity. So now we will move on with the discussion for coenzymes and cofactors.